So today we start a new chapter titled Receptor-Mediated Endocytosis. So what we've been doing in the last lecture or last few lectures was receptor ligand binding. So we are essentially looking at the chemistry of receptor ligand binding, the kinetics, and how it could be a linear kinetics, a simple linear kinetics, or a bimolecular kinetics, and so on. So now what we want to do is we want to study the main process of not of receptor ligand binding, but what receptor ligand binding leads to, what is its functionality in the body. And so one of the major processes that receptor ligand binding is involved in is known as receptor mediated endocytosis. Now I'll talk about endocytosis, what it means, and so on, all the details. So so that is the whole idea, apply whatever we had learned in this new chapter and apply whatever we had learned in the old chapter about receptor ligand binding kinetics to the process. And the process itself that we are concerned with here is receptor mediated endocytosis. Okay, so let's go to the slides and look at it. So this, as I said, the chapter is titled receptor mediated endocytosis. So what is the definition and function of receptor mediated endocytosis? We need to understand that. So what we do in endocytosis, forget receptor mediated, just endocytosis itself, is that it's a major pathway by which macromolecules can enter the system. And you see I have underlined the word macromolecules here. What am I trying to say over here? We had studied previously in another course that how micromolecules, how small molecules enter the cell, right? So there are transport processes, ion channels, passive diffusion, ion pumps, and different kinds of co-transport, and that is transport in different directions. One is you have a carrier mediated transport, so the carrier will take one of these. So these are all processes through which small molecules enter the cell. Okay, micromolecules enter the cell, and these are natural processes which are part of the cell transport mechanism. Now if you want to transport a macromolecule, a very large molecule inside the cell, how do you do that? Because the cell doesn't have an intrinsic or inherent mechanism to do that. And the process to transport macromolecule inside the cell is known as endocytosis. You have another process which is opposite of that, and that is the process of transporting macromolecules out of the cell, and that's known as exocytosis. Okay, so receptor mediated endocytosis. So endocytosis is a major pathway by which macromolecules enter the cell, and receptor mediated means that endocytosis which is mediated or facilitated by receptors. Like you have carrier mediated transport for micromolecules, for small molecules, here you have receptor mediated. So that's a major difference between, say, one of the things that that's similar akin to receptor mediated endocytosis is carrier mediated transport. So what are the major difference? Receptor mediated endocytosis is for macromolecules, and carrier mediated transport is for micromolecules. That one is carrier mediated, where one of the carriers essentially binds with the protein or whatever you want to transport, and then transport it inside and dissociates. Here also there is a similarity, almost a parallel correspondence to that. That is, one of the receptor itself, you can think of the receptor as a carrier which binds to the ligands, bring the ligands in, or the whatever you want to transport, the macromolecule which is the ligand, brings it in and then throws it back. Now we are going to study the process in detail of what is this process of bringing it in and then throwing it back into the cytoplasm. We are going to study this process in detail, but all I am trying to say over here, there is almost a 1:1 correspondence between carrier mediated transport and this one. Only two differences: carrier mediated transport transports micromolecules, receptor mediated transport transports macromolecules, A, and B is that carrier mediated transport is much simpler. The mechanism is a lot simpler. Here you will see that the mechanism is a lot more complicated. There it was only dealing with chemistry. Here it is chemistry and a lot of physical involvement, because the macromolecules are large. So you need ways to entrap those macromolecules, which is very different. You follow what I am trying to say. So because the micromolecule is small, so you can just bring it in, but because the macromolecule itself has a volume, so there are ways to entrap this. And what we will give special attention to is that what if the macromolecule is a bacteria or virus that you want to, macromolecule is a virus or a bacteria that you want to kill. So what do you do then? So that is another thing we are going to look at. So the second point over here is that receptor mediated endocytosis acts to internalize. This is how you kill a bacteria or virus. It internalizes the protein or whatever is the macromolecule is, doesn't have to be necessarily protein, and generation of signals, and for the regulation of receptors numbers. So let's go one by one. to these 3 points. So, first one is internalization of proteins for the metabolism. So, as I said that macromolecules are larger molecules, and there has to be a mechanism to bring this inside the cell. It is not as simple, the mechanism of bringing this inside the cell is not as simple as it was for carrier mediated transport, where it just simply reacts. So, there is a mechanism, and this mechanism is called the process of internalization, and we will study this in much more detail today itself. The second one is a generation of signals. And as I said that receptor mediated transport or receptor ligand binding in itself plays a very vital role in generation of signals, in neurotransmission. So, transmission from one part of the body, say between the brain and another part of the body, and it has a huge impact in, and I discussed this in one of the earlier slides here, that the impact it has on, or function it has in different kinds of neurotransmitters. As you see, these are receptor mediated, these are receptors, the one that you see on the left, epinephrine, serotonin, acetylcholine, the first 3 for example. So, they are neurotransmitters. So, they help in connecting 2 distant parts of the body through cell signaling. Okay. So, cell signaling is something like telephoning, so each one part of the body which is far away from the other part of the body, so they are sort of telephoning each other and talking to each other and telling that, okay, I need this, more of this. It's like two business people talking to each other and say, so I need more of this supply of this chemical, and somebody saying, okay, no, I have got too much of it, I need to throw out. So, there is this whole talking that is going on, and the central nervous system is sort of nodal point there, So, the central nervous system is, so if one part of the body which is very distant from another part of the body has to talk to each other, then it has to go to the central nodal point, which is like the telephone exchange out there, which is the central nervous system. So, it sends the neurotransmission, it neurotransmits to the brain, and then the brain will neurotransmit back to another. So, these neurotransmitters and hormones, so these are some of the examples, and so this is the cell signaling part. So, this receptor ligand binding plays a very vital role in cell signaling. Another thing that I had talked to in the previous lecture or the lecture before, I think, was about T cells, that immune cells for example. So, when there is immune attack on your lung or some other part of the body, how would that part of the body inform that to the brain that there has been an immune attack on the system, right? So, there also cell signaling is important. So, there are certain cells, for example, in the lung there are T cells which will relay this message. So, there are these messengers. So, they will relay this message, but where does receptor ligand binding play a role? It is that whenever, for example, there is an immune attack, so certain ligands, okay, in this case say a bacteria or a virus, is going to bind to the receptor, and as soon as the receptors, those receptors that are there, so for that particular virus or bacteria, so those are sensor receptors that are there. So, they will bind specifically to a bacteria or virus, and when they bind specifically to a bacteria or virus, then they immediately, so these are like spies out there in the lung. So, immediately they send a signal back to the central nervous system of the brain that some bacteria or virus have been caught here, and it is very likely that there is a immune attack on the system. Then the brain sends signal back to the lung saying that yes, there has been an immune attack. So, if not just one, maybe 2 or 3 of these T cells send these message across that there has been an immune attack on the system, then what happens is that the the brain says yes, there has been it believes that there has been a total immune attack on the system, and it needs to start the immune response, which means that we can talk about that a little later. Immune response means essentially that the thing would be geared up, the whole immune system would be geared up, or in other words, what it means is that it goes from the resting state to the activated state. These are biological terms that are used. Okay, and the comparison is like, as I said, between from peace state to the war state. So, an enemy has attacked the land and or the lung, and the system needs to go from a war state. Okay, so whenever the nation is in a war, so the war state mentality is different from peace state mentality. In biological terms, these are called not peace and war state; they are called resting state and activated state. So, the system goes from the resting state to the activated state. So, this activation process is also done through cell signaling. Okay, and everywhere there is a cell signaling, and cell signaling has become a very major part of biological studies these days, including in chemical engineering. So, everywhere there is a cell signaling, receptor ligand binding plays a very important role. Okay. So, these are the 3 points I think that we are talking about. The protein internalization of the protein, explain that. generation of signaling, which is cell signaling, and the third one is the regulation of the receptor number, and this is equally important. What happens is that the receptor number in the cell can change. Okay. So, till now in our earlier calculations, if you remember in the calculations that we did in the last chapter, we always assume that the receptor number to be a constant, okay, nRT to be a constant, but in reality that is not true, because within a cell or within any particular organ the receptor numbers are changing. The reason they are changing is they are dying, or they are being produced, whatever is the case. So, receptor numbers are changing because of that. Now, when these receptor numbers are changing, there is a necessity for the cell to regulate the receptor number. Say for example, receptors are dying and they are continuously decreasing. Now, what will happen? Two things will happen. The ability of the cell to do a process like endocytosis will decrease, that is metabolism. So, endocytosis is very important. See for example, you have a very large macromolecule, a protein or something, a carbohydrate, a large macromolecule which cannot enter the cell. Okay, glucose can enter the cell of course through carrier mediated transport, but much larger than that. You want it to, forget the immune part of it, just the fact the metabolic part of it. So, you want that macromolecule to enter the cell. It is not possible for the macromolecule to enter the cell, and the example that we will do is lipoproteins, okay, through the rest of this chapter. These lipoproteins are very large molecules, okay, and you can have large sugars also, which if you cannot internalize them, then it can lead to diabetes. So, the one that we are going to look at is lipoprotein, and these are large molecules. There is no way to internalize these large molecules without being able to without the physical internalization. There is no other way of bringing this, transporting these very large molecules, because ion pumps, these are not ions, molecular channels are not going to work. You cannot do carrier mediated transport because this is too large for a carrier mediated transport. So, the only way you can get this inside the cell is through the process of internalization, and the process of internalization is therefore important for metabolic activity, right. So, that is one part, and again the cell signaling is the other part. So, the regulation of the receptor number, as you see over here, the last point, is important for both these activities. So, if the receptor number decreases beyond a point, then what will happen? The internalization of the proteins that you want to do is not going to happen, A, and if the receptor number decreases a lot, then you cannot have cell signaling also. So, both these it is important to regulate. Now, what happens if the receptor number goes up a lot? That is also no good either, right. So, if the receptor number decreases, we understood what would happen. If the receptor number goes up a lot, that is no good either. The reason being that you will have too much internalization of proteins, it will be beyond the levels that a cell can take, or your system could be too sensitive to any kind of cell signaling. So, you have a message, just one message, and you are not supposed to send that message back to the brain, and you send that message back to the brain, and a whole immune response could be triggered off where it is not important. And an example for this is people who have problems with tonsils. Okay. So, what are tonsils? Do you know what are tonsils? Tonsils are essentially the immune cells. Okay, they are a bunch of immune cells. So, lymph nodes, lymphoids, and their essential function is immune action. So, they are essentially the first level of immunity even before it enters the lung. So, when you say that somebody has a problem with tonsils, what is meant is that you have a hypersensitive tonsil. Okay. So, tonsils, the main job of the tonsil is to sense that there has been an immune attack on the system and send that message to the brain. That is all the tonsil is supposed to do. And if you have a hypersensitive tonsil, what happens is that, like I got, so what happens is that it sends a message back to the brain. You do not have a problem essentially. There is not much of an infection attack. Infections are there everywhere. You are breathing in infection all the time. So, maybe a very trace amount of infection, but the tonsil senses that and thinks that there is a huge immune attack on the system, and it sends a message to the brain, and the brain starts the whole immune response. So, you start to get fever, your immune system is enhanced, you go from the immune system goes from the resting state to the activated state, and so on. All this entire process starts off in the tonsils. So, what would happen? Essentially that what would happen is that when you have a hypersensitive tonsil, on the surface of the tonsil, on the cells of the tonsil, you probably have an excess of. So, when somebody has a so-called tonsil problem, so it means that probably there will be an excess of these receptors, signaling receptors, so which will trigger off the entire signaling mechanism. So, as a result, what you need to do, you need to regulate the number of receptors. You do not need it too high, you do not need it too small. So, this is the thing that I just mentioned, that the macromolecules. So, the molecules transported by these mechanisms so include nutrients. So, as I said that it is not just immune, saving the system from immune attacks and virus and bacteria, but they include nutrients, and this example I gave, low density lipoprotein LDL, immunoglobulins, insulin, so that is why it is so receptor ligand binding is so important for diabetes, and growth factors as well as viruses and bacteria. Okay. So, this is one part of it, the immune system part of one part of it, and then these nutrients, these are all important for the growth and metabolism in the body, the LDL, the insulin and so on. The example that we will take in this chapter is the example of LDL low density lipoprotein. Okay. So, this is a picture, I think you can see it hopefully properly. Can you see this properly? Okay. So, this is the picture for LDL, the low density lipoprotein, what happens. So, I have better pictures, let us just have a quick look at this. So, this is a LDL molecule, a low density lipoprotein molecule, which is a large macromolecule which cannot enter the cell otherwise, and you have these LDL receptors out here. Okay. So, these blue things are the receptors. So, this is how you always represent a receptor, a cup like structure with a stem out there, and ligands are always represented by dots. So, when they combine, you represent them as so this is the ligand with the dot, and this, and then combined form would be represented as a cup like structure plus with a dot on it. So, even when you draw your pictures, this is how you are supposed to draw. So, this is the plasma membrane, and as I said before that the receptor is such that a part of it, the stem, is inside the cytoplasm and the cup like structure is protruding out of the cytoplasm, and this is for to capture. Now, this is called a coated pits, and I will come to that a little later. This protein structure that you see over here is called a coated pit. Let us not worry about that at this point of time. Let us look at the simple thing. So, this lipoprotein comes and binds with the receptor. Then this coated pit that you have over here forms what is known as a vesicle. Okay. And I will talk about that, how this vesicle is formed and all that stuff. We will do this in details. So, forms what is known as a vesicle. Then the vesicle coating goes away and you have this uncoated vesicle, and so this thing grows a little bit and you have this endosome. Now, in the endosome, and I will discuss this, what happens is that there is a pH change in the endosome. As a result, so whenever the receptor ligand binding process is taking place, what is the reaction? L plus R giving. So, this receptor ligand binding, for example, L plus R giving complex, right. So, in the vesicle you have this complex, right. So, the coated vesicle that you have and that gets uncoated, you have the complex. But remember, you have this K plus, let us call this, and let us call this K minus. So, you have these two rate constants, and K plus and K minus are functions of pH, okay, of temperature also and pH, but here pH is what is important. So, what happens is that the pH, the endosome, so if I can go to the screen now, inside the endosome, here you have here the complex form. Now, inside the endosome the pH changes, typically they decrease the pH, they acidify the endosome and decrease the pH. Why they do it is, if you decrease the pH, then the backward reaction is facilitated. Okay, you have the K plus, the forward reaction rate constant, you have the backward reaction rate constant. So, the forward reaction rate constant is facilitated or enhanced at higher pH, whereas the backward reaction rate constant increases at lower pH. So, if the pH of the system is lowered, then the backward reaction happens, right. So, then what happens? The complex dissociates preferentially into the ligand and the receptor. So, this is what has happened. So, you see inside this in the picture, so the complex is dissociated into the ligand and the receptor, and then you form this lysosome, what is basically, so you have internalized. Now, what has happened? This LDL was outside the cell, now you have been able to internalize this, and this is broken down and all these things, and the receptor is sent back. Why is the receptor sent back? Because the body is a very miser thing, it does not want to waste anything. So, as I said, it needs to regulate the number of, so this receptor sitting inside the cell cytoplasm makes no sense, it is not going to function, it is not going to do its job. So, it has to be sent back to the cell surface where it will continue doing what it is doing. Okay. So, I hope this is clear, right. So we will come back to it. I have better pictures and more detailed discussion. So let's look at the mechanism of endocytosis. I already explained that. It's just going point by point. Everything that I said is what is written here. So through the interaction of the cytoplasmic protein, cell membrane components from vesicle and transport form vesicles and transport membrane fluid and solids from one region of the cell to another. Fine. There's no big deal out here. So endocytosis, that's the definition of endocytosis. It's a process of vesicle formation from the plasma membrane. So you have this process of vesicle formation out here is known as endocytosis. So outside it was there, it entered, and that step is known as endocytosis. Exocytosis is a process by which the membrane vesicles formed from Golgi apparatus fuse with the plasma membrane and release newly synthesized proteins to the extracellular fluid. We are not studying exocytosis in this chapter, but this is just to inform you what the definition is. It is a reverse process. So if you synthesize something inside and you want it into the bloodstream, so this is the process, just the opposite process of endocytosis. And endocytosis can result in turnover of plasma membrane lipids every 1 to 10 hours at a rate much faster than membrane synthesis. Vesicles do not diffuse in the cytoplasm, but are transported along a network of microtubules. So what this first sentence means is that if you go back to the picture, this process of the membrane turnover, that is the membrane being recycled in and out, that can happen over 10 hours. So with endocytosis, the membrane itself over a period of 10 hours can be recycled in and then out. Okay, so the whole membrane recycle process. And vesicles do not diffuse in the cytoplasm, but are transported along a network of microtubules, which means that once a vesicle has been formed over here, it is not going to diffuse from one part of the cytoplasm, but there's some microtubules that are formed inside, through which the channel-like structures through which it can flow. Okay. So this is, I think, important, and this will be necessary what is up there now for our analysis. So classification of membranes involved in receptors involved in receptor-mediated endocytosis. So there are two kinds of receptors that we typically talk of. One is called a class 1 receptor and the other one is called a class 2 receptor. Let's go to the definitions and I'll explain in the next few minutes. So class 1 receptors are receptors for hormones and nutrients, and these receptors bind to the ligands before binding to the coated pits. Let's just go through the definition now and then explain in a minute. And class 2 receptors are receptors like LDL. These bind to the coated pit without the ligand, and the ligand receptor binding follows that. Okay. Now let's see what this is. So this picture that you see over here, maybe I have another picture. No, yeah. So this picture that you see over here, see, this is a receptor. These are the receptors. Okay, that are there, and then this receptor binds to the ligand. You see that where the arrow is. So these black dots are my receptor, and my receptor goes and binds to the ligands. The green things are the ligands here, and then the coated pits are formed. Okay, and let's see, maybe we have the other way around here. No, it's the same kind actually. I'm sorry, but anyhow, so this receptor binds to the ligands before binding to the coated pit. So this is my class 1 receptor, and what would be my class 2 receptor? Would be where it first forms a coated pit and then it. So there's very little difference, but up to that. So whatever difference we have is up to this point 2, number 2. Beyond that, it's all the same. The receptors and the class 1 and class 2 receptors are all the same. So you have the binding process over here. So it forms the receptor ligand complex, and then these complexes then diffuse laterally, and this is one thing that is slightly different, and we'll come to that in the next lecture or the lecture after that, is the diffusion of these bound things, the complexes. So when one of the major differences is that when the system binds to when this receptor like complex, in case of class 2 receptors, when the coated pit is first formed and then the complex is formed, then the lateral diffusion will take place for the complex itself as a whole. Okay. Whereas in this case the lateral diffusion would be the lateral diffusion would take place for the coated pit with the complex. Okay. So the coated pit with the complex will move laterally, fine. Whereas here it would be the individual complexes that have to move together if there is to be a lateral diffusion, followed by the formation of the coated pit. And this is a these proteins come together, and we will study that later. These proteins come together, the Raptor protein and Cla3 and Danamine, they come together to form the coated pit. So the coated pit is essentially like a thicker part of the membrane, like kind of thickens, another layer of, you can think of that another layer of protein adds to the membrane and thickens the membrane. Why is that done? Why do you think that is done?
Cell ensures that between any bacteria or something doesn't diffuse inside.
Yeah. So that's one part of it. Essentially, that when you form the vesicle, vesicle, you want to make sure that it is mechanically stable. There is mechanical rigidity for the vesicle, and the vesicle does not rupture. And as he said, that if the vesicle ruptures, then what will happen? If there is a virus, these are nutrients, then no problem at all. But if there is a virus, then if the vesicle ruptures just like that before the bacteria is killed, then the bacteria will actually go and infringe into the cell, and whatever you wanted to do, you will have the reverse effect of. You wanted to kill the bacteria, but you are in a way facilitating the bacteria to enter the cell. Okay. So that's why you form a structure, a protein structure, just to give stability to this entire vesicle, so that it's not ruptured at any point of time, unless the cell wants it to rupture. Okay. And as you see this whole process, you will see that there is an inherent wisdom of the cell. So the cell is doing things in a way that it really wants to do. So look here. So then the vesicle is formed. Okay. And then the coated pit is thrown out and recycled, so that it can go back to the membrane, because the coated pit doesn't have to be there all through, and this goes back over here and forms on the surface. Why? So that it can be there to trap in the next set of to facilitate the next set of receptor ligand binding. Fine. So this is now the uncoated vesicle, just as we showed, and then this process of transformation happens, that is the pH of the system changes. Now how do you think the pH of the system would change? This vesicle so from here to here, the pH is obviously changed. Otherwise, what you see over here is that the dissociation has occurred. The receptors have unbound themselves from the ligands, and that cannot happen, as I just now explained, that cannot happen unless the pH of the system changes. So how do you think the pH of the system changes? Yes, exactly. So what happens is this: on these vesicles, they open up hydrogen pumps, okay, hydrogen ion pumps, and these hydrogen ion pumps that are opened up pump in hydrogen ion from the outside, from the cytoplasm inside the vesicle, and when these hydrogen ions are pumped in from the cytoplasm inside the vesicles, the pH of the system goes down, and the reaction is facilitated or favored in the backward direction, and the complex now dissociates into the ligand and the receptor. So here, after this sort of dissociation takes place, then now it's time for the ligand and the receptor to separate out, and as you can see over here, the ligands go back and stick to the membrane of this vesicle, while the receptors are encapsulated within, and then it forms these vesicles, and the ligand forms a separate part of the so receptors form a separate vesicle, the ligands form a separate vesicle, and the ligands go back to the cell membrane to do its next cycle of transport, okay, because you have a limited number of receptors and you don't have the ability to waste them. And these ligands, as you can see here, they could be used for different kinds. They could be used for metabolism, if they are proteins or nutrients that you need. If they are not needed, then they could be thrown out, and we'll do a detail. So I hope this process, step by step process, is clear to all of you. The reason I am saying is because we have to do a detailed modeling of all these steps of the process, and unless the physical mechanism is not clear to us, it is not possible. So if there is any question on this, you can ask. This is the same thing, essentially the same kind of picture, that the receptor ligands bind to the receptor, and then the coated pit is formed, so class 1, and exactly very similar kind of thing. The coated pit goes away. I mean, I don't think there is any point in repeating this one, right? Okay. So now let's go step by step to the process. I think I explained most of them, but let's still walk through this. So, step 1 is this, because each of these steps, when we model the system, we have to understand and quantify each of these steps. Okay. So, that is the whole idea. So, in receptor mediated endocytosis, first is the binding of the protein, which is a ligand, to the specific receptor. So, these are specific receptors, and we had discussed this at the beginning of the receptor ligand binding chapter, that there is a whole issue of specificity. So, one kind of receptor is supposed to bind to one or another two kinds of different ligands. So, the binding of the receptor to specific protein to the specific receptor, and the receptors migrate into specialized regions in the cell membrane known as coated pits and identifiable by dark membrane in the TEM. So, coated pits typically cover a few percent of the cell membrane. So, this again, you know, if you should realize that here we are again talking of which class of receptors? Class 1 is what I showed you, where the ligand binding, receptor ligand binding takes place before the coated pits are formed, but here we are talking of the coated pits being formed first. Okay. So, this is class 2. So, because the reason we are looking at class 2 is essentially we are planning to model the class 2 kind of receptor, and I discussed this. The specific example that we are going to talk of is low density lipoprotein, and that is a class 2 kind of receptor. So, just small changes at the beginning of the, so the entire process is more or less same, just a small change. So, accordingly you have to vary your model initially. So, if I ask you to do the class 1 kind of modeling, so just a small change at the beginning, and then the rest of the process is going to be more or less same. So, this is my picture. So, these are the this is the coated pit, and this shows as dark dark edges in the transmission electron microscope. And they cover a few percent up to 5% of the cell membrane, not a very large amount of the cell membrane. So, coated pits form continuously, form coated vesicles every 2 or 3 minutes. So, so this is the time scale of the system. So, once a coated pit is formed, the total receptor ligand formation and the vesicle formation, that total process takes around 2 or 3 minutes, the cycle of that, the internalization process. Okay. Then internalization has already happened. Within a minute or two after the internalization has happened, this protein and the cytoplasmic protein that is present, clathrin, dissociates from the vesicle, and the vesicle is now referred to as the early endosome. Why is it called the early endosome? How is it different from the late endosome? Is the pH factor, okay, and as we just discussed. Now the whole question of pH comes in. So, early endosome, as you see over here in the picture, early endosome will have the receptors bound to the ligands, okay, whereas this is the late endosome where there will be a dissociation, and how that dissociation is effected is through the hydrogen ion pumps. So, at this point what happens is ion pumps in the vesicle membrane pump hydrogen ion to lower the pH within the endosome. Okay. So, the ion pumps open up and hydrogen ion streams into the vesicle, and the pH of the system is lowered. And because of the lowering of the pH, the receptor ligand affinity decreases with the decreasing of pH, or the forward rate constant decreases and the backward rate constant increases, and the complex now starts to dissociate into the receptor and the ligand, and that leads to the formation of the late endosome. This is the late endosome. Okay. So, 5 was the formation of the late endosome, where the receptors and the ligands are dissociated and segregated, not just dissociated and also segregated. Segregation, how it would help? Because now they have to split into 2 different parts. So, if they are segregated, spatially segregated, the splitting would make sure that one of the endosome contains all the ligands, one of the endosome contains all the receptors. So, most ligand molecules remain in vesicles that are directed to the lysosomes, where proteins are degraded. If these are proteins, they would be degraded to amino acids, and from the time a ligand molecule binds to a receptor or finds a receptor, it takes around 15 minutes to reach the lysosome. So, this whole process that we are talking of, let us see, from here all the way to here, this process is around 15 minutes, and this entire process is around 20 minutes. Okay, that is the time scale of the entire receptor ligand binding process, and the formation, what I said, from here to the formation of the endosome, which contains, so from the vesicle, the late endosome, and then the formation of the endosome which only contains the ligand, that process takes around 15 minutes. Okay. So most types of receptors are sorted and sent to the tubular structures that return the receptors to the cell membrane. So, let's go back to the picture. So these receptors that are there, these are microtubules that are there, that are formed, and these microtubules together send the receptors back to the cell membrane, where they will be ready for the next set of receptor ligand binding. And LDL receptors, this is LDL, as I said, 20 minutes, but it's not strict for each kind of thing, it will be different, but that's the time scale. So LDL receptors make this whole round trip, the receptors, in 12 minutes, and each receptor makes 150 round trips before degradation. Okay, so this is a receptor, 12 minutes. The endosome and the ligand, it will take a little bit more time, because the ligand formation, because the receptors leave a little early, if you remember. So maybe 5 minutes more or something like that for the ligands, but the receptor makes a round trip in 12 minutes, and each receptor does 150 round trips. So what happens after 150 round trips? These receptors essentially are degraded, they die. Okay, fine. So this is a process. Now next thing we are going to do is there any doubt or question on the process itself? So the next, because if we understand the process, the next thing we are going to look at is the kinetic studies on the process. So the reason and the thing that we are going to focus on is receptor low density lipoprotein, as I said, LR binding for low density lipoprotein. And the reason we are doing this, so LDL receptor. So a few words on LDL low density lipoprotein. So these are the ligands that would bind to the LDL receptors. So when we talk of, I think LDLR is going to be the symbol that we'll use for LDL receptors, and LDL for the protein, I think that's what we use. So LDL low density lipoprotein plays, why do we want it? Plays an important role in the delivery of cholesterol to the cell. The LDL molecule itself consists of a core of cholesterol ester surrounded by cholesterol, phospholipids and proteins. I think I don't have that slide with me now, but we had at some point of time shown a picture earlier of how this looks like, with the cholesterol sticking, the phospholipids and the proteins and the cholesterol molecules sticking out, if you remember, and that was in the previous course. Anyhow, maybe I'll get that slide and show it to you in the next class. So LDL, and this is something we had already discussed. LDL, the internalization process, is a class 2 by class 2 receptors, which means that the coated pits are formed first before the binding occurs. And what we need to understand is the dynamics of the LDL receptor, because this dynamics of LDL receptors forms the basis for the genetic disease which is known as familial hypercholesterolemia. Okay. This disease is characterized by extremely high levels of plasma cholesterol and the early onset of cardiovascular diseases. So what happens in this disease, familial hypercholesterolemia, as I said, we can, there are several diseases which have to do with LR binding, diabetes, as I just talked about, in the insulin, which is also a class 2 thing. What happens in this case, for example, this familial hypercholesterolemia. So let's look at the terms, each of the terms. Familial hypercholesterolemia. So when you say these two, it consists of 3 words: familial hypercholesterolemia. Okay. So first thing that we're talking about is you're dealing with cholesterol molecule. That's why it's the word cholesterolemia. Hypercholesterolemia. Okay, excess of cholesterol. And familial means it's a genetic disease. So 3 points: it's a genetic disease which involves excess of cholesterol. But my question to you is, excess of cholesterol where? In the bloodstream, yes. So what is happening? It is not that, so people who, and this is a genetic disease that we have already said, so people who suffer from this disease, say familial hypercholesterolemia, it's not that they're intaking a lot of fatty things. Even if they reduce their fat and keep it to a very normal level or even below that normal level, still then it's not going to make much of a difference. What is happening is that these fat, these low density lipoproteins, which are important for the cell, they have to be internalized. There is no other way. These are macromolecules. The only way they can enter is through this internalization process. And this internalization mechanism is something that has gone kaput in these people. That is not what is not working, this internalization mechanism. So as a result, whatever cholesterol they intake end up being in the bloodstream. Okay. And that is why, I think sometimes we don't understand when we say high cholesterol, we don't understand where the cholesterol is. The point that I'm trying to make is that the cholesterol is in the blood. Okay, the cholesterol remains in the blood, whereas it should actually be going to the cell and going through the inside the cell. It should go through a metabolic process. So that is being hampered, and it remains inside the cell, inside the blood. Sorry. And what happens when it remains inside the blood? It leads to cardiac diseases. Can any of you think of why it leads to cardiac diseases? How would you do that? Yeah, so basically what happens is that, so these are my arteries, say, if you're looking at the artery from the top. So these are the membrane of the artery. These cholesterol molecules, they are sticky. Okay, they are sticky. So what happens is they start sticking to these arteries. Okay, like this. So they're supposed to go inside the cell, but because they cannot go inside the cell, they are roaming around, and they start sticking to the membrane of the cardiac vessels, the arteries, the capillaries, whatever, especially the arteries. And then they reduce the diameter. Now when you reduce the diameter, what would happen? Blood pressure will increase. Okay, blood velocity will also increase, and the pressure will also increase. So essentially the all kinds of kinetic form of energy that is there in the bloodstream will increase, and as a result you have what is known as ischemia, right? So ischemia. Okay, so you have what is known as a ischemic heart. So ischemia is basically small heart attacks that happen, and this can lead to large heart attacks when large arteries. So small heart attacks are like when small arteries are being ruptured, and then you can have a bunch of these small arteries rupturing at the same point of time. That's one possibility, or a large artery rupturing. So whenever this ischemia would happen, inferior ischemia, this is called in medical terms. So whenever inferior ischemia would happen, this arteries would rupture, and because there's extreme high pressure, the blood will put too much pressure on them. This goes back to your entire fluid mechanics and everything that you have studied, right? So what will happen is the shear stress will increase on the surface, right? Because your velocity is high, and the distance between the distance. What will happen is the velocity itself is increasing, and if you look at this, let's say, this itself. So let's say the velocity here is something v bar, and the velocity here has to be 0 from no slip, right? So and this is your r. Okay, so del v del r is your gradient. Let's say this is r bar. So in this case, del v del r would be an average, approximately v over r bar, right? So what is happening is your velocity is increasing and your radius is decreasing, both ways, as a result of which your gradient is increasing, velocity gradient is increasing, both because the velocity itself, the mean maximum velocity is increasing, and also the so this is actually half of, average velocity is half of that. So gradient is half of v over r bar, average gradient. So both your average velocity is increasing and your diameter is decreasing. So because of both these effects occurring simultaneously, your shear stress is increasing. So shear stress, when it's increasing, it leads to enormous stress on the membrane. So you can ask, one of the clever questions you can ask me is that it might be harder for the vessel to rupture, right? Because you are thickening it. Okay, so that's there, but the point is that so much the shear stress increases so much on the membrane that at one point of time the membrane ruptures and heart attack happens. So that is the whole mechanism of heart attack. I am not sure if you have studied this before, but this is the whole mechanism of heart attack when we talk of it. And so it is important. That is why this disease becomes so important, because it's very, and I think that's somewhere that Indians are genetically predisposed to this disease. South Asians, not just Indians. That is why South Asians are asked not to eat a lot of fatty material. Because if you compare with people in the West or people in the South, say Africa or in the Western countries, they eat a lot more fatty material than Indians do. But Indians have a hugely prone to heart attack risk. South Asians, I think, more than 50% of all the heart attacks that happen are actually of South Asians. Okay, and that doesn't have to do with the fact that we have we eat a lot of cholesterol. We actually eat a lot less cholesterol than other people do. That has to do with the fact that this is a genetic disease, and we are genetically predisposed to this disease because the receptor ligand binding mechanism that is there for this low density lipoprotein or lipoprotein in general for us, it's not good for South Asians, and that is something when we do the kinetic study, we'll try and understand that what makes us genetically predisposed to this particular disease. Okay. So, several mutations, so that brings directly to the point that I am talking about, that several mutations of the low density lipoprotein receptor can produce this disease. Okay, and let's look at what these mutations are, and then later on we'll try and connect this with the kinetics of the process. So, first case is that the affinity between the low density lipoprotein and this receptor itself is low. Okay. So that Kd increases, leading to impaired binding. Kd is what? The dissociation constant, or in other words the backward rate constant over the forward rate constant. Now, one possibility, so why are we South Asians genetically predisposed? And as I said, it has to do with the kinetics of the receptor ligand binding, not because of the fact that we eat too much fat, which we don't, not because of the fact that our arteries are all smaller than everybody else's, no, which is not, but because of the fact that the receptor ligand binding process is not favorable for us, and it is not favorable because of some kinetic issues. So, the first issue is that the binding rate constant between the receptor and the ligand for people who are genetically predisposed is low, or in other words the dissociation rate constant is large. The second possibility is that no, the binding is actually all right, but it is the internalization process. So, maybe the coated pit is formed, maybe the binding occurs as it should, but the internalization of the coated pit is not happening properly. Maybe the coated pit is not favoring internalization, or the internalization mechanism, whatever is the kinetics of the internalization process, is not favorable towards the process of internalization, right. So, this is a completely distinct possibility. The first possibility is that the binding is not happening properly, and the second possibility is no, the binding is happening all right, but the internalization is not happening all right. The third possibility is that this internalization is not a problem, the binding is not a problem either, but what is the problem is that several rare mutations involve normal binding to Ldl receptors, but little localization in coated pits. Okay. You see what I am saying. So, let us go back to the picture. Yeah. Sorry, though this is the first class 1 receptor, but still I will try to explain with this picture. See, what is happening is that maybe the receptors are binding all right to the coated pit, maybe the internalization is all right, but within the coated pit not enough of those bound receptor ligands are localizing, because unless they localize, see, if the bound receptor ligand is here or there or somewhere else, it makes no difference. It has to localize on the coated pit for a vesicle to be formed. Is it clear, Pallavi? Okay, clear to everybody? So, it has to localize on the coated pit for the vesicle to be formed, because if it does not localize on the coated pit, the vesicle is not going to be formed. So, though the coated pit formation process might be all right, but the localization itself is not right. So, excuse me, these are the three major mutations that can happen. One is the affinity, second one is the defect in internalization, and third one is that the coated pit itself is forming all right, but the localization is not happening all right. Now, why do you think that the localization of these receptor ligands to the coated pit might be hindered? See, think of a class 2 receptor, do not think of a class, this is where the differences come in, do not think of a class 1 receptor. See, class 1 receptors are easier, because what happens is that you have these surface and you have these things over here, and then once sorry, this is not bound yet, and then the coated pit forms over here, okay, and then the binding occurs. So, now but here you have the bound things over here, and then the coated pit is going to form.
The lateral movement.
Right, exactly. The lateral diffusion. So, for a coated pit to be formed, they have to come in close proximity to each other. These bound ligand receptors have to come in close proximity to each other, okay, and therefore it is necessary that they move close to each other. Okay, so from here I need to go to a place where it is something like this, okay. And then the coated pit can form, because there is no, it's not possible for it to form a coated pit as long as this. A, because the proteins necessary for formation of such a large coated pit is not available, and B, if you form such a large coated pit, then the vesicle is going to be really huge. You know, the cell cannot afford to have such a huge vesicle, because you have to provide the environment, you have to increase the pH and decrease the pH and all that stuff. So all these mutations together, or any one of them or any two of them, some permutation combination of these mutations, provide a net effect that the number of functional LDL receptors that can undergo binding and internalization is reduced, and this reduces the LDL uptake. And as you can see that all these mutations that we are talking of are generally, you know, these are mutations or genetic mutations, and therefore the disease is genetic, okay. So it is not, so one of the things we understand is that, you know, when you go to a doctor, anybody who has a high cholesterol or the diabetes, what is the first question the doctor asks you? That did anybody from your father's side or your mother's side had this disease, both for diabetes and for LDL cholesterol, high cholesterol. So that's the first question they ask. Did any of your parents or grandparents have diabetes or cholesterol? Same with, you know, anybody who has cancer. One of the questions that the doctors ask is that, oh, what, you know, whether anybody, there's a family history of cancer in cancer. So because these diseases, you know, and especially the diseases that we are considering, hypercholesterolemia or say diabetes, these are diseases which are resulting from receptor ligand binding, which is exactly a genetic process. So that's why it can happen, and the possibilities are these three, okay. Now we'll do this in the next class, but just to give you an overview, that so what we want to do is, why do we need to quantify, why do we need to do this, is, as I said, that this is a genetic process and there are 3 mutations possible, okay, which can lead to 3 kinds of defects. Now if I want to understand or quantify these 3 defects, then I have to do a total kinetic study and then look at the kinetic constants for each of these processes, kinetic or transport, whatever is the case. Maybe the lateral diffusion could be a transport process, and we'll talk about that later. So these 3 processes that are there, and we have to quantify that, and then we'd be able to quantify the disease in general. So the kinetic process includes LDL protein binding, internalization, degradation, as well as identify the effects of mutation on the process, okay. So these are, I think I'll stop here, but just let me brief you. So these are the 3 things that we are going to look at. So the binding, internalization and degradation, and added to that would be the thing that, you know, he just mentioned, which is the formation of the coated pit and the lateral movement, because we have to make sure that when we make a model, all these 3 genetic mutations that cause this disease are included in the model, okay, which is the dissociation and the internalization and the migration of the bound receptor ligands to a localization, so to say, so that the coated pit is formed. So all these 3 we'll try to incorporate into our model, and we'll do the model in the next lecture.